Good evening, everyone, and welcome to tonight's lecture. I'm George Kraw here with Raquel Chamorro-Garcia. This series features acclaimed UC Santa Cruz scientists and technologists who are grappling with some of the biggest questions of our time. Tonight, we learn how our environment and pollutants in our environment affect our own health and the health of future generations. Our speaker, Professor Raquel Chamorro-Garcia's research revolves around deciphering the mechanisms which exposure to environmental pollutants during pregnancy and other critical windows of susceptibility contribute to increased predisposition to disease and obesity in offspring and unexposed descendants of multiple generations. Before we begin, I would like to share a few details about the event tonight. We are using a webinar tool, so there's no chat function. We will have an opportunity to answer our questions at the end of the program, and we invite you to submit your questions into the Q&A box at any time. Tonight's event will be recorded and be available on the UC Santa Cruz YouTube channel. Our conversation tonight will be moderated by Susan Strom. She is a distinguished professor of molecular cell and developmental biology at UC Santa Cruz. Susan earned her BA in chemistry at the University of New Mexico and her PhD in biochemistry at the University of Washington. She did her postdoctoral training at the University of Colorado, where she began studying germ cell development in the model organism C. elegans. She moved her lab from Indiana to UC Santa Cruz in 2007. Her research team studies mechanisms that specify and protect the identity, immortality, and proper development of germ cells. The team's current focus is on epigenetic regulation of germ cells across generations. Professor Strom, thank you for moderating the program for us.
All right. Thank you, George, for that introduction. And thank you, George and Rafe, for hosting these Kraw Lectures. They have been much needed and appreciated opportunities for us to share sort of exciting updates on our questions and advances, especially during the last 2 pandemic years. So thank you for bringing these to us. Our Kraw lecturer today is Dr. Raquel Chamorro-Garcia. She's an assistant professor in the Department of Microbiology and Environmental Toxicology, METOX. I vividly remember hearing her job seminar in 2019. She presented important and fascinating findings that George already alluded to and that you'll be hearing more about in Raquel's talk. I was excited that she got the job offer from UC Santa Cruz and even more excited that she accepted it and joined us in 2019. Her research investigates how a mother's experiences, her diet, her lifestyle, exposures to chemicals and other environmental insults can affect the health of her descendants for many generations, even out to great-great-grandchildren. That's amazing. She's using mice as her model organism, and I think you'll understand why when you hear her lecture. She was trained in biochemistry, both her BS and her PhD, at the Autonomous University of Madrid in Spain, and then she moved into her current research area when she went to UC Irvine to do her postdoctoral research. Her current research program here at UC Santa Cruz lies at the intersection of 3 currently hot areas: epigenetics, metabolomics, and transgenerational inheritance. As I said, she and her lab are carrying out their experiments in mice where they can ask directed questions and can ethically do experiments. But she hopes in the near term to take some of her findings in mice and apply them to diagnosing metabolic diseases in human populations. So I will turn the screen over to Raquel to present her Kraw lecture. And like George, I invite you all to type your questions in the Q&A box during the talk and after, and we'll come back to those during discussion afterwards. Raquel, your turn.
All right. Well, thank you, Susan, for that nice introduction. And thank you, organizers and the Kraw family for inviting me to present here today the research that we do in my lab. So I'm going to share my screen so we can start. All right, I hope everybody can hear me and can see my presentation. So as George and Susan mentioned, my lab is interested in understanding how environmental chemicals are contributing to metabolic disruption and more specifically obesity. And this is important because metabolic diseases are becoming a very wide global problem. So just to give you an example regarding obesity, this map is representing the prevalence of obesity back in the mid-70s, where around 10 to 15% of the population worldwide was considered obese. And obesity here is defined as a body mass index when this value is equal or larger than 30. If we flash forward to 2016, we can see that the situation has changed very dramatically. So now there are many countries in the world, pretty much all countries in the world have increased prevalence of obesity in their populations. And in some cases, like in the United States, over 40% of the population is now considered obese. So obesity accounts for 3.4 million deaths, obesity and overweight, 3.4 million deaths every year around the world. It is an increasing problem because it is not only seen in adult population, but also in children. And there is a strong association between obesity during childhood and obesity later in life. And one important point is that as of now, the number of countries succeeding in decreasing obesity in the last few decades is basically zero. So obesity is also important because it has like a wide ranging costs, including economic costs. So it is estimated that around $2 trillion annually are spent in treating obesity worldwide, and that includes direct costs and indirect costs in disability. It is also associated with social costs because, for instance, developing countries cannot afford the cost of treating obesity. There are significant racial disparities in obesity prevalence, and there is a lot of stigma around obesity. So it is important that we understand what are the contributing factors to obesity and other metabolic diseases. So of course, genetics is an important factor when there are alterations in genes like leptin or insulin that are very important in regulating different metabolic processes. We can see obesity during adulthood. But now we also know that the environment can also play an important role. We know that excessive consumption of calories and not exercising to burn down those calories can contribute to obesity. And more recently, microbiome, stress, and chemicals found in the environment have been shown to play an important role in obesity during adulthood. However, there are other moments in life where exposure to certain factors can play an important role in developing obesity later in life. Oh, something that I forgot to say is that genetic factors only explain less than 3% of the cases. So there are other factors, as I was saying, regarding environmental factors that are playing a role here. So as I was saying, there are other moments in life where exposure to certain environments can contribute to disease later in life. So one example is in utero development. So smoking or poor nutrition during pregnancy can contribute to obesity in the offspring. And now we also know that the environment our ancestors were exposed to may be playing a role in obesity trends currently. One example in human populations is the Dutch famine. This famine occurred at the end of World War II when there was a shortage in food availability in the Netherlands. So here on the right, I'm showing a graph where this column on the left is representing the desired number of calories that every adult should be intaking every day. Of course, during the war, those numbers were reduced because food availability was reduced. But by the end of 1944 and the beginning of 1945, there was a dramatic drop in the calories taken by the population. And something that is very extraordinary about this study is that there are really good records about the health of the population in this region during that time, and more specifically about the health of women that were pregnant at that time. So something that was found is that the exposure to famine during preconception and during in utero, during pregnancy, in this Dutch population led to adverse health outcomes in the offspring. And the health outcomes that were found were very different. There were some of them that were related to metabolic disorders like obesity or diabetes, but there were also cases of infertility or neurological disorders. And in general, there was an increase in adult mortality. And something that was very important was that the records not only were well maintained in the offspring, but also in their offspring. So this would be like the grandchildren of the women that were exposed to the famine back in the 40s. And in general, this offspring also has poor health. So how does this happen? How can we explain this multigenerational inheritance of disease due to the exposure to a famine? So when we think in the traditional way, we think about genetics. So in that context, we will have an environmental perturbation that will lead to one mutation in our DNA. And that mutation will lead to one particular phenotype following Mendelian laws. However, what was seen in the Dutch famine cohort was that there was an environmental perturbation that would be the famine that led to alterations that are still yet to be determined. And the phenotypes were very different and very variable. So in this context is when epigenetics comes to play. So what is epigenetics? There are many different definitions for epigenetics. So I'm not going to get into which ones are better than others depending on what, but I'm just going to stick with this one that states that epigenetics is the study of changes in organisms caused by modifications of gene expression without altering the DNA sequence. So gene expression refers to the process by which the information that is carried by a gene is transcribed into the gene product that is called the messenger RNA. And this gene product is translated into proteins, and the proteins are the ones that are leading to one particular phenotype or appearance. So with epigenetics, we are able to regulate the gene expression, so the information carried by the genes, without altering the DNA sequence. This means without introducing any mutation in the DNA sequence. So I would like to use an analogy to explain what epigenetics is and how it works regulating the expression of the genome. And I'm going to talk about music. So when we think about music, we have the music pentagram and the basic notes. And these basic notes are going to sound slightly different depending on how you use the notation. So depending on how you use this notation, you can create a beginner's version of a traditional song like Happy Birthday to You, or you can create a more complex, advanced version of the same song. So when we bring biology into this, the music pentagram will be the genome, the basic notes will be the genes, the notation will be the epigenetic marks that are going to regulate how the genes are being expressed in the genome. And that can lead to a healthy liver cell, for example, that is working properly and regulating the metabolism of the individual. Or depending on how the epigenetic marks are used, it can lead to an unhealthy liver cell. So it's still a liver cell, but it is not working properly. So what are these epigenetic marks? When we start learning about our genome, when we are in middle school or in high school, we always start learning about this DNA helix, that it is like the more simplified version of how the genome is represented in the cell. So this DNA helix contains the genes, and it can wrap around a family of proteins that are called histones. These histones and the DNA helix form chromatin, and the chromatin can wrap around itself, making more complex structures like chromatin loops, chromatin domains, and chromatin compartments. And this bubble here is representing the nucleus of the cell, and these little tangle cartoons here are representing the different chromosomes. So it seems that this information, these chromosomes are located in the nucleus in a very random way, but there is actually a little bit of a structure there, and I will get into that in just a second. So epigenetic marks can be placed at different levels in this structure. So we have DNA methylation, that it actually means that the methyl groups are going to be placed in different locations in the DNA helix. And this DNA methylation have the potential to silence or make more accessible the expression of certain genes. There are also modifications that can be introduced in the histones. And there are also alterations that can occur at higher order nuclear architecture. So this means that there are alterations on how the genome is structured within the nucleus. And as I said, this seems like a random way of distributing the genome in the nucleus, but there is a little bit of a structure there. There are two compartments that are can be identified, compartment A and compartment B. Compartment A is represented here in green, and it is generally rich in euchromatic regions. And this means that those regions are more open to being transcribed. So this means that the genes that are in those regions are have a higher probability of being expressed. And in general, euchromatin is located in the center of the nucleus. Compartment B is represented here in red and is rich in heterochromatic regions, and the genes that are located in those regions are generally silenced. There are other elements of epigenetic components that can regulate gene expression, and those are non-coding RNAs, and I'm not going to talk too much about it right now. So what is the role of environment in human disease? I would like to introduce you to the term the exposome, which refers to the measure of all the exposures of an individual in a lifetime and how those exposures relate to health. So we often think about our exposures as a single measure, like we are exposed to one diet or one chemical or one individual factor. However, our environment is very complex, and we need to understand that there are we have exposures that come from at different levels. So factors about our ecosystem or where we live can contribute to our health. Our lifestyle choices like diet or physical activity are also important contributors. Our social environment, like our household income or social capital or social networks, are also important factors in our health. There are other factors that are perhaps more commonly known, that are the physical and chemical factors, and they include like a long list of different factors. So all these factors interact with each other, so they don't act on our bodies or on our genome independently. They can interact with each other, increasing the risk for certain diseases. And if this is not complex enough, they also interact with our genome. So depending on what is our genomic makeup or epigenomic makeup, we are going to react differently to the exposure to these factors. So my lab is particularly interested in chemical exposures. and more specifically on endocrine disrupting chemicals, or EDCs, that are defined as exogenous chemicals that interfere with any aspects of hormone action. So how do these chemicals work? So in our endocrine system is in charge of regulating very important biological functions like reproduction or metabolism. And it does so because of the existence of these molecules that are called hormones, that they send signals in our bodies, and they regulate all these biological processes. So endocrine disrupting chemicals, what they do is to actually interfere with the activity of the hormones, and therefore they can alter these biological processes, leading to disease. So the first one of the first reports of endocrine disrupting chemicals having an effect in wildlife was published by Raquel Carson, and it was in this book, Silent Spring. And this was back in the 60s. In the 90s, Theo Colborn and collaborators were continuing the work of Raquel Carson, but they were focusing on the effect of these chemicals in human populations, and they published this book, Our Stolen Future. And after that, there have been many other books that have been discussing the effect of these chemicals in our health. Excuse me. So my lab is interested in understanding how endocrine disrupting chemicals are contributing to multigenerational metabolic disruption. So my first contact with this project was when I started my postdoc back in UC Irvine, and I started working with Tribulatine, or TBT, which is a chemical found in the environment. It is used as an antifouling agent in paints and wood preservatives. So this is just an example of what can happen if the paint that you use in a boat doesn't have Tribulatine. However, the use of Tribulatine is very well regulated because it has been shown to have a very detrimental effect in marine species. It is also a thermal stabilizer, and it is found in house dust and human blood. So this suggests that humans are exposed to Tribulatine. In the lab, we have seen that TBT induces lipid storage in cell culture, and it also induces fat tissue development in mice. So this suggests that the original findings suggested that TBT was contributing to perhaps obesity if humans were exposed to it. So my question started by asking whether TBT induces transgenerational obesity. So I did this experiment in 3 occasions, and I was able to replicate the results. I'm just going to give you an overview of my findings. So in this experimental design, what I did was to expose pregnant moms, and we were working with mice, to TBT. And then we tracked multiple generations to look for any effects in metabolic disruption, including increased adiposity as a marker of obesity. So with this particular experimental design, when we are exposing pregnant females, we are also potentially exposing the first generation, the children that are developing in the uterus, but also the second generation, because these fetuses that are developing have primordial germ cells that are going to turn into the mature oocytes and sperm that are going to lead to the grandchildren. So the first generation that can be considered unexposed are the great-grandchildren, and any generations after that. So in order to address whether TBT has a transgenerational effect, we need to look into the great-grandchildren and future generations. So what I found was that ancestral exposure to TBT led to an increase in accumulation of fat in the great-grandchildren and great-great-grandchildren, larger fat cells, which suggest an increase in lipid storage. and increased fat content in the liver, which suggests non-alcoholic fatty liver disease. So we actually challenged our animals with different diets to actually evaluate their metabolic status. So we generally keep the animals on a standard diet, and they have ad libitum free access to food. They can eat whenever they want to eat. And what I did was at a certain point, I switched the diet to a high-fat diet for a few weeks, and then I switched back to a standard diet. So this could be equivalent to human populations depending on the season we are at and the diet that we are taking. So one example could be during the summer or fall, we are taking a balanced and healthy diet, but then the holiday season comes and we eat perhaps not so healthy diet, not so healthy food, and then in the winter and the spring we go back to a healthy diet. And this increase in calories or this access to not such a healthy diet can have a very important detrimental effects in our health. So in this experiment, what we found was in the males, we measure percentage fat content. So this is important. We were measuring fat content, not body weight. And we found that when we switched the diet to the high-fat diet that is represented here, the animals that were ancestrally exposed to TBT, that is this dotted line, accumulated significantly more fat more rapidly than the control animals that are labeled here as DMSO and represented in the solid line. When we return the animals to the standard diet, the animals ancestrally exposed to TBT didn't fully recover. So that this suggests a permanent change in the metabolism of these animals. When we looked into female data, we didn't see statistically significant changes, but there is a trend towards the same results as we found in males. So this data is kind of suggesting that at least in males, the phenotype is not fully revealed until we expose the animals to a high-fat diet. However, something that we found was that before the diet challenge, we measure all the metabolites that are in the blood in these animals. So I'm going just to explain to you what these graphs represent. So when we measure all the metabolites in the blood, there are many different metabolites. So it's very difficult to actually represent all the metabolites and their level of expression in all the animals that we were studying here. So this type of representation is a way to reduce the complexity of the data, and they take all the information from all the metabolites and generate one single value for each of the mice. So the closer these stars are from each other, the more similar the metabolic profile is in between these animals. So here I am representing in green the DMSO group, which will be the control, and then two different concentrations of TBT represented in light and dark blue. So as you can see, all the animals that belong to the control group are clustering very close together, which indicates that they are very similar among each other. And the same happens with each of the TBT groups. And the most important factor here is that they separate from each other. So this means that each group is having a very unique metabolomic profile, or there is a very unique way on how the metabolites are being expressed in the blood. In females, we see a similar pattern. There is a really good separation among the groups, but there is good clustering within the groups. So this actually suggests, if you remember in the previous slide, I'm sorry, females didn't show a significant increase in fat content when they were exposed to the high-fat diet. but they are actually showing a very distinct metabolic profile even before the diet challenge. So this means that both males and females are being affected by the exposure, the ancestral exposure to tributyltin. So how does this happen? Something that we wanted to do is to actually understand what is the mechanism of action for TBT to lead to metabolic disruption in the great-grandchildren and the great-great-grandchildren. So we dived into the molecular mechanisms. And to do that, we looked into different tissues of these animals. We took fat tissue, liver, and mesenchymal stem cells, MSCs. Mesenchymal stem cells are the precursors of the fat cells. So it was a way for us to look into the cells that are going to become the fat tissue. So we looked into males and females. And what we did was to look into altered patterns of DNA methylation that, if you remember, is one of the epigenetic marks that can be altered and can contribute to differences in gene expression. And we also look at a whole genome expression. So this means we looked into every single gene, the level of expression of those genes in each mouse. So I'm not going to go into a lot of detail about how we reached to the results, but I'm going to show you basically the conclusions. Our conclusions were that TBT, ancestral exposure to TBT, introduces changes in higher order chromatin organization, which means that refers to how the chromatin is structured within the nucleus. These alterations of higher order chromatin organization will subsequently introduce changes in DNA methylation, and these will actually contribute to alterations in gene expression. So questions that are currently open in my lab, all the data that I just showed you was work that I did during my postdoc, and it built the story that I am continuing now as an independent researcher. Excuse me. So current questions that are open in my lab refers to what are the critical windows of susceptibility to environmental exposures, which is actually, I think, an important question to actually determine in which stages during our lifetime the exposure to these chemicals is going to have the most dramatic impact. And in this case, it will be a negative impact in our health. Other questions are related with how these alterations are inherited and whether different environmental factors follow similar mechanisms of action. So what are the critical windows of susceptibility to environmental exposures? I want to go back to the Dutch famine example. So as I mentioned, the data that was found that the women that were exposed to the famine during in utero during pregnancy, the offspring will show adverse health outcomes. But something that was important, it was that there was really good records about the health of the offspring, depending on whether the famine occurred in early, middle, or late gestation, and even during preconception times. And the data suggested that preconception was a critical window of susceptibility. So in those women that were exposed to the famine before getting pregnant and during the very early stages of pregnancy, their offspring showed a more dramatic adverse health outcome. So this brought into question what would be the effect of tributyltin exposure during preconception periods. So basically what we are testing in the lab is to actually, we are exposing the females before conception, and then we are tracking, the goal is to track multiple generations, but for now we are just tracking the effect in the children. So what we found is that the animals whose mothers were exposed to TBT only during preconception had a significantly increased body weight. So this graph is representing the age of the animals here at the bottom, and this is represented in weeks. And here, I am just going to tell you anything that is above the 0 line means that there is it is an increase in body weight in the TBT group compared to the control group. So this data is showing that in weeks 5, 6, and 7, and 8, the animals that whose mothers were exposed to Tribulatine before conception had an increased body weight. One limitation of this approach I want to bring up, the graphs that I showed before regarding the diet challenge when we were switching the diet to a high-fat diet were focused on fat content. So we were able to actually measure fat content in the animals. In this case, we don't have access to the instrument that is necessary to look into the difference between body weight, fat content and lean content. So we were basing our analysis in body weight. So that can be a limitation because body weight can be affected by many factors, not only fat, lean content, bone, hair, water content can also play a role in total body weight. So this is at the level of body weight information, but something that we also did was to take liver samples and to look into the expression of the genes in the liver. So here on panel B, the left panel is representing the data from males. And just to, without going into a lot of detail, just want to say that anything that is above the 0 line again is significantly overexpressed in the TBT group compared to the DMSO group. And anything that is below the 0 line is underexpressed in the TBT group compared to the DMSO group. So this y-axis is actually representing different regions of the genome with different GC content. And different GC content can also refer to the different compartments in the genome. If you remember, I talked about compartment A and B and how they are enriched in euchromatin or heterochromatin. So basically what we were seeing here is that the animals whose mothers were exposed to Tribulatine had a bias towards how the genome was expressed, depending on whether the genes were located in one region, heterochromatic here, that has low GC content, or in a different region, rich in GC content. So this suggests that there is actually an alteration or higher order chromatin organization, and that is affecting how the genome is being expressed. In females, we see the opposite pattern. So the regions with low GC content were underexpressed in animals whose mothers were exposed to TBT compared to the DMSO, the control animals. And the regions with high GC content were overexpressed in the TBT animals. So this is also indicating that there is a sexually dimorphic response to the preconception exposure to TBT. So this means that males and females are responding to TBT, but they are responding to TBT in different ways. So how are these alterations inherited? One of the things that we are looking into right now is to actually look into what is happening to the germline. So if we are treating during preconception periods to the exposing the moms during preconception periods, our hypothesis is that environmental chemicals can be affecting the oocyte before fertilization. And those alterations can be carried after fertilization, and that can actually affect how chromatin organization is established in early stages of development. Other projects that we are exploring right now are include studying the effect in primordial germ cells, or PGCs. So these cells will become the future oocytes and sperm. So we would like to know what is happening in these cells to actually predict whether there is going to be a propagation of the alterations from one generation to future generations. Other questions that are on the table right now refer to whether different environmental factors follow similar mechanisms of action. So as I mentioned earlier, we are exposed to many different factors, and they interact with each other and they interact with our genome. So factors that we are interested in are arsenic, tobacco-related chemicals, different diets, and of course, chemical mixtures. But because we are exposed to many factors, analyzing one factor at a time is only going to give us partial information. So chemical mixtures is actually a very important topic in our field. We are not, I'm not going to show any data about this, but it's a direction that we would like to follow in the future. And I'm going to talk about inorganic arsenic. So inorganic arsenic is a global problem. There are many different places in the world where the concentration of arsenic in drinking water is higher than desirable. So arsenic exposure guidelines by the World Health Organization states that levels equal or lower than 10 micrograms per liter of arsenic in the water can be considered safe. However, there are some hot spots worldwide where there are higher concentrations of arsenic in the drinking water. And I just want to zoom into the United States, just to give you a sense of the levels of exposure in different places. So this map is actually representing the arsenic content in drinking water in different places in the United States. And there are many regions where the concentration is way above the 10 microgram per liter safe level that the World Health Organization is stating safe. So what is known about inorganic arsenic and disease? So it has been associated with skin lesions, neuropathies, gastrointestinal diseases, metabolic disruption, and so on and so forth. The routes of exposure are mainly via drinking water, but there is also exposure during industrial processes. We are exposed through food that has been watered with water that contains arsenic. And smoking can also be an important source of arsenic. There is a little bit of information about the modes of action. So it can alter DNA repair, contributing to alterations on how the genome is being expressed. It can contribute to oxidative stress and alter the phenotype of stem cell populations. The modes of action that I am more interested in is related to DNA methylation. And again, this relates with epigenetic alteration of epigenetic marks in our genome. So we did the same in this experiment that I explained a few slides ago, in which we exposed females during preconception periods to TDT. We also had a group that was exposed to arsenic. And what we found in terms of body weight was also like some trends towards increased body weight in animals whose mothers were exposed to inorganic arsenic. And when we looked into the expression of the genome in the liver, we also found a bias depending on where the genes were located in the genome. Remember, regions with low GC content are associated with heterochromatic regions, and regions with high GC content are associated with euchromatic regions. So this effect is more dramatic in females than in males. And in this case, the pattern is more similar between males and females compared to the patterns that we observed in TBT. So this data suggests that arsenic is actually following a similar mechanism of action to lead to metabolic disruption in the offspring. So what is next? One of the next steps that we want to pursue is to actually dive into what is happening in human populations. So all the data that I just showed you was based on work done in mouse work. So does this relate to what we are seeing in human populations? So this is a tricky question because there are certain things that we cannot do with human populations, as you can imagine, but there are some information that can be taken from the human samples that are available. So we are actually working with a group, and we are hoping to get funding. We are waiting to hear from a grant to actually use human data, human samples, and capitalize in our methodology to analyze DNA methylation and look into as a proxy to determine whether there are alterations of higher order chromatin organization. So we are the group that we are collaborating with is led by Brenda Eskenazi in UC Berkeley, and also Asa Bretman from UC Merced. So they launched this study that is called the Tamakos study. That is, Tamakos stands for Center for the Health Assessment of Mothers and Children of Salinas. They started this project back in the late 90s, and they were actually studying pairs of exposed pregnant women to different chemicals found in the environment. Most of these women were working in farms, so they were exposed to pesticides, and they tracked the health of the children, and this has been occurring in the last 20 years. So this is a very powerful study that can give us very significant information about whether exposure to these pesticides can be contributing to adverse health outcomes in children after exposure during in utero development. And something that I didn't mention is that these children tend to show metabolic disruption in their phenotypes. So we hope that with the study of these human samples, we can actually develop diagnostic tools to determine susceptibility to metabolic diseases. And that can actually be important to predict and prevent metabolic disruption in future generations. And with that, I would like to thank everybody in my lab, my collaborators, and my funding, and I will be happy to answer any questions.
Thank you so much, Raquel. I told you it would be fascinating. I didn't tell you, you already knew that. We have lots of questions in the Q&A box. I'm going to start by asking you to tell the listeners the time between exposing a mother and analyzing her great-great-grandchildren. Because we think about why would mice be a model? And one reason is because it all happens fast. So what is that time? Is it 2 days, 2 weeks, 2 months, 2 years?
So each generation in mice takes more or less 3 months. So in an experiment where we are actually studying 4 generations, it takes a year from the moment we start exposing the pregnant mothers to the moment we are analyzing the great-great-grandchildren. So it is a short period of time compared to human populations. If we wanted to go to human populations, it will take several decades. And it has a number of problems to work with human populations because you need to keep track of the descendants, and you want to make sure that the descendants agree in participating into the study that you are working on. Of course, working with mice is a lot faster, but there are other animal models that are even faster than like mice. Like one example will be worms or fruit flies. So there is not a perfect model, but this is the model that we have in the lab.
Great. Let's see. How do you know the impact of TBT or other chemicals is not an indirect impact through the microbiome or other secondary pathways?
So we actually don't know. I mean, that can definitely be one factor. So we, with the analysis that I showed in the in my presentation, we were actually analyzing target tissues. So we are now trying to track back, to step back a little bit, and to see what is the origin of these alterations. The hypotheses that we are considering right now are related with alterations in the germline. However, we cannot discard the possibility of having TBT altering the microbiome of the mother during pregnancy, and that can affect the health of the offspring. And it is an important question because when we are working with mothers, during pregnancy, the effects that we are seeing in the offspring, we don't know if they are due to direct exposure of the chemical over the developing embryo, or if the chemical or the environmental factor that we are working with is affecting the maternal physiology, and those alterations affect the development of the embryo. So all those questions are on the table in my lab, and we actually recently got a little bit of funding to address part of those questions, to actually explore what is the role of the maternal physiology in this equation.
So one way to avoid the maternal physiology issue is to ask whether exposure of fathers has transgenerational effects. You're not exposing the whole oocyte, which is what the embryo, develops from. And I think you could be looking at effects straight away in children and grandchildren, and certainly go out multiple generations. Have you or others looked at exposure of fathers and transgenerational effects?
Yeah, there is actually a significant amount of work done in the last maybe 15 years related with paternal exposure to environmental factors. And one of the persons that is working on that is Upasna Sharma, which is in UC Santa Cruz. So that is an important question right now. And what has been seen is that by exposing males, the fathers, to certain environmental factors, there can be alterations that occur in the sperm. And then that can affect the offspring after fertilization. So that is definitely an important question to ask, because we put a lot of pressure on when we talk about humans. During pregnancy, the mom is always taking care of herself, just to make sure that there are no risks to the baby. But with the preconception studies, either the female or the male preconception studies, that puts the pressure even before pregnancy, and actually the responsibility falls in both parts of the equation. So yeah, it is an important question. There are people working on that, and we are actually studying a new study that is also exposing males to factors related with tobacco smoke, and tracking what is happening in future generations.
Great. So you showed in at least one slide that pregnant mice were exposed to various concentrations of TBT.
Mm-hmm.
Were the higher doses shown to lead to higher percentage of fat content? So was it dialable amount of TBT and fat?
So yeah, I briefly mentioned that I did this experiment in 3 different occasions. So I ran 3 different experiments, and we used 3 different concentrations in the first experiment, and we tracked multiple generations, and we don't see a dose response, a linear dose response, which is relatively common when we talk about endocrine disrupting chemicals. It is not uncommon to see non-monotonic dose response, which means that when we are looking at one particular trait, we don't expect to see the more concentration, the more response. At some point, we can actually lose that effect. And the reasons why that happen are, it can be many different explanations depending on the mechanism of the chemical that we are interested in. So what I found with the three, when I used three concentrations of TDT, was that there was not a linear dose response in every single trait that I looked into. In some cases, we saw that response, and fat content was one of those cases. But when we were looking at other traits, we didn't see that linear dose response.
Okay. Several of our participants asked about the surprising finding that males and females show a different liver response. I think it's transcriptome of the liver after preconception exposure to TDT. What do you hypothesize underlies that sexually dimorphic response?
Yes, so we don't know yet what is happening, but males and females are different. So that is the first, the rough answer to the question. So males and females are different, and they are different at the genomic level. So our hypothesis is that tribulatine is altering how chromatin is organized in the nucleus. And I briefly talked about the euchromatin and the heterochromatin and how it defines two different compartments. So the male genome has a Y chromosome that is very heterochromatic. Mm-hmm. And that can affect the proportion of heterochromatic regions or how the establishment of the chromatin architecture occurs in the nucleus. The female nucleus has two X chromosomes and they are not that heterochromatic, so the distribution of the heterochromatin regions can be variable. Hmm. So one hypothesis that we have is that whatever occurs, whatever alteration occurs, it is happening very early on after fertilization, and it is because of the presence or absence of a Y chromosome in the zygote.
Interesting. Why focus on just DNA methylation and not histone modifications? That did not come from me. Someone else.
Yeah, so the DNA methylation, so this project started a few years ago because it takes a long time to develop these, generate these materials. At the time that when we were starting this project, the hypothesis was focused on DNA methylation. So the field was focusing more, the field on endocrine disrupting chemicals were focusing more on DNA methylation mechanisms of action. So we started looking into DNA methylation we prioritized that instead of histone modifications. When it comes to the connection with human studies, we want to focus on DNA methylation because in order to do histone modifications, you need to preserve your material in a very specific way. So then you can actually analyze histone modifications. When we are talking about human samples, it is very unlikely that you are going to have the material preserved in that way. And actually, you need a lot of material to actually do histone modification analysis. So we are... with the DNA methylation, we can actually use very, very little amount of DNA to actually look into what is the methylation profile of the DNA. So originally, the intention wasn't to actually translate that into humans, but it's actually turning out very nicely for us to actually make associations with what is happening in our mouse model compared to what we can study in human studies.
So related to DNA methylation, What are the group of genes where altered methylation occurred? Are they involved in a specific process or processes like metabolism?
Yes, so we looked into that, and there is an enrichment in genes involved in metabolic processes, and also genes involved in chromatin structure processes. So we are not only seeing that there are alterations in the genes that can explain the phenotype that we are observing in the mice, but we are also seeing alterations in genes that can explain the mechanism of action that we think is occurring in the cells.
Okay, great. This is an interesting question. Are you seeing positive impacts of chemical exposures on longevity of offspring or health rather than always adverse effects?
So that's an interesting question. And actually, yeah, I think it is very interesting because we always tend to focus on the negatives.
Yeah.
And actually, you know, we get a lot of questions about, okay, there are chemicals that are inducing obesity or that are associated with metabolic disruption and obesity in a negative way. But are there chemicals that can induce the opposite, to help metabolizing fats better or to... so the answer is that there is very little information about that because the focus tends to be, at least in our field, in aspects that relate to human disease, so we can actually get funded. So there is not a lot of information about that. And I think it's a very interesting question. And actually, I have talked about this with people in my lab in many occasions. But, you know, these type of experiments are very expensive, and you want to make sure that you have funding to do them. And the public agencies are generally more interested in studies that are going to help understanding better human disease.
Yeah. So I'm looking at your lab gift fund, and I'm thinking the pandemic probably set back your research, not being able to get to your mice. And I'm wondering, what do you need to speed up your research program?
Yeah, that's a great question. And actually, indeed, yeah, the pandemic has been very dramatic for everybody, but starting the lab in 2019 and all of a sudden closing the lab in 2020 for a few months was very impactful. But so I think, you know, something that I briefly mentioned in my talk is there is this instrument that allows us to analyze fat content in our animals. It's called the Echo MRI. And I had access to one Echo MRI when I was a postdoc, and it was very powerful because it will allow us to measure fat content, which is one of the important endpoints in my research, to address whether chemicals are having an impact in adiposity and obesity. And one important factor is that you can do longitudinal studies without sacrificing the animals. So you will put the animal in the instrument, and in just 2 minutes, you will have the data and you will have like fat content in the animals. Since I don't have that instrument right now, I cannot do these longitudinal studies. So the only way that I have to measure fat content is to sacrifice the mice and then weigh the fat depots that we isolate from the animals. And that reduces a lot the power that we have to actually do interventions, like, for instance, decide if we want to introduce a diet challenge or if we want to do any other type of intervention to evaluate the effect over the fat content of the animal. So it would be great to have access to one of those. And I'm actually actively looking for either public or private funding to actually buy one of those instruments.
Very good. All right. In the late 1960s, a classmate bragged to me about a wonderful new technology that his father's farm used, a growth hormone injected into livestock. This increased by 25% how fast animals could pass through the feedlot. Growth hormone use in livestock was and perhaps remains widespread in the US, where obesity is prevalent. Are there studies showing this to be associated with obesity?
So there are, and that actually increases the complexity of all the questions, because on the one hand, we have the effects of the hormones that were used to increase adiposity in the animals, and how that affects, when we eat that meat, and that might be higher in fats. It also has the problem about us being exposed directly to those hormones that are present in the tissues of the animals. And so I'm saying that it increases the complexity of the study, because there can be confounding factors. There is the component of the fat that comes from the animal, and also from the hormones and other factors that are injected in the animals to actually increase production or stock in farms.
Okay. So this is a comment and then a question. This does not sound like good news to reversing the epidemic of obesity, especially in children. What can you project from your work? What can we learn from regions where there is little or no obesity? And I might tack on to that. What about these school programs where the schools are trying to feed children healthy lunches to try to set them on a better path and maybe reverse some of the trend that might come from eating perhaps junk food?
Yes, so there is so those are very important questions to ask. So to answer the question about regions where in the world, what was the first part of the question?
One was, what can we learn from regions where there is little or no obesity? That's one.
So to answer that question, so in the maps that I showed at the beginning of the talk, there is actually evidence that there has been an increase in obesity prevalence in pretty much every country in the world. And the projections are actually showing that that is not going to change. And is actually being, that growth is expected to be larger in those regions of the country, of the world, where there is, as of now, lower cases of obesity. So the projections are there. In terms of the effect of a healthy lifestyle during childhood, something that happens in research when we work in developmental biology, we always see development as a critical window of susceptibility, which is a very critical window of susceptibility. However, after we are born, there is a period until adulthood where there is a lot going on in our bodies, and the exposures that we receive during that period can have a long-lasting effect, not only in our health, but also in our descendants. So having healthy habits like eating healthy or exercising more during childhood can also really help preventing adverse health outcomes later in life. And linking back to the microbiome, we have a microbiologist in my department that works in microbiome, and he always says that he thinks that the earlier we start eating healthy, and this conversation came talking about how my kid that is 1 year old right now, when he started eating solid food, he was saying that it was very important that he started eating healthy as soon as possible, so his microbiome was going to be better, and that was going to help in preventing certain diseases later in life.
Good advice. That's Michael Patnode, right?
Yes, yeah, Michael. Yeah.
Excellent. Have you considered the epigenetic implications of virus infections and vaccinations of great-great-grandparents?
I haven't thought about it. I don't have an answer to that, actually.
How many exposures and of what duration result in genetic alterations? So you talked about exposing preconception moms or pregnant moms. How long is the exposure? And do you have to do it repeatedly?
Yes, so yeah, something that I didn't mention is that we do all these exposures in drinking water. And we do that to mimic kind of like a natural exposure as opposed to injecting the chemical, which is a lot more aggressive. And but if we are injecting the chemical, we can control the amount the animals have taken. So there are pros and cons with every approach, but we decided to follow the drinking water approach. So the animals could drink as much or as little as they wanted, and that could actually represent better human exposures. So in terms of the length, pregnancy in mice, it takes 3 weeks. So when we expose the animals during pregnancy, it will last 3 weeks. And we will, depending on the chemical, we will change the water more or less frequently. And this is because some chemicals are not very stable. So we will have to put fresh water with fresh chemical in the water to make sure that the active compound is reaching the tissues or the embryos that we are targeting. In terms of preconception exposure, what we are doing with females is actually track and males. We are actually using the number of weeks that takes for in the case of females, for an oocyte to mature, and that will be 6 weeks. So we will treat the animals for 6 weeks with the goal of affecting the process of maturation of the oocyte, and then we will mate the animals. In the case of males, we do the same, and that exposure, I think it is 5 weeks. 5 weeks, yeah, 35 days, 5 weeks, and we will expose them in drinking water, and then we will mate the animals. Yeah, the goal is to actually target the maturation of the germ cells.
Got it. Okay. Could effects of increased obesity in non-directly exposed subsequent generations be due to obesity in one or both parents? That is, might obesity be a self-perpetuating phenotype? Does that make sense? That obese parent, I think that, another way to say it might be, if obesity, are obese parents more likely to have obese children regardless of the cause of the obesity? You're looking at obesity caused by TBT.
Yes. So you are referring to the parental physiology playing a role in the next generation disease. That is,
I think
the
so.
way
I'm reading the question.
You're reading the question. Yeah. So, well, I am understanding the question that way, and definitely there is evidence in human studies where that is actually the case, that obesity in the parents can contribute to obesity in the next generation. So yeah, that is definitely a confounding factor that is not to take into account.
Yeah. So how long does the epigenetic effect persist in your system? I mean, you've already gone out to great-great-grandchildren. And so I guess the question is, how far can you go out? Is your lab ever up for that? Do later generations eventually revert to normal? And then I might tack on to that question, but I'll let you answer that one first. Do you have any sense whether the effect is passed through the sperm or the oocyte once you get past that first exposed mother? So first question, how long does the epigenetic effect persist in your system? So
in my studies, the farthest that I got was the 4th generation. So that will be the great-great-grandchildren. And we have actually seen the effect in that generation. As I said, working with mice doesn't take as long as working with human cohorts, but it's still quite a lot of work. And this is also something that I have talked to people in my lab, and it will be great to actually have the funding support to actually do this type of studies to maintain the generations for a long time. So we could actually track whether the effect disappears. There is information in, there is work done in worms. Patrick Allard from UCLA ran a study with BPA, bisphenol A, which is also an endocrine disrupting chemical. And he actually found that after 7 generations, there was actually a dilution of the phenotype. So it can be that we lose the phenotype after several generations.
Yeah.
In terms of how that relates to humans, it actually, 7 generations is a lot of time. And one important factor related to human populations is that we are constantly being exposed. So unless we remove all the chemicals from the environment, we are always going to potentially have the impact of chemicals that are contributing to whatever phenotype we are looking into. So,
and furthermore, human populations are mobile. They move around, and so kids may leave the place where they were born, move to a whole nother region, and then you have a whole nother set of variables.
Exactly. Yeah.
On top. Yeah.
Yeah.
Well, then my question tacked onto that was, do you have any sense whether the effects you see are transmitted through the sperm or through the oocytes of children, grandchildren, great-grandchildren? I'm not talking about the exposed mother. That's obviously the oocyte. But after that.
So the way my experiment is designed doesn't address that question. So basically what we do in order to produce the following generation, what we do is to mate animals that were exposed to the chemical. We avoid mating siblings, so, to avoid synergistic effects or increasing the effects that we are seeing just because we are mating siblings. So we avoid mating siblings that we mate within the same treatment. So in that context, we cannot tell if the phenotype, if the inheritance occurs via the maternal germline or the paternal germline or both. In order to do that, we will have to do outcrossing. So that means that in each generation, we will take one treated or ancestrally treated mouse and expose them to opposite sex non-exposed animals and then track the offspring. So in that case, we can actually see if the phenotype is carried by the father or the mother. And that could be a way to do it. And there is research done in that direction. Michael Skinner has done some work in that direction, and it is definitely something that is also interesting to do. There are so many things that we could do, and that is interesting to do, but there is basically no time to do everything. So we are trying to narrow down the burning questions that we have right now and start answering them as we go.
Good. Are there changes in telomere length with your toxic exposure?
Can you say that again?
Are there changes in the length of telomeres, the ends of chromosomes?
I haven't looked into that, but it's definitely something interesting since we are looking at heterochromatic, the differences between euchromatic and heterochromatic regions and the distribution of those regions in the nucleus. It will definitely be interesting to look into that and to see if that is can actually be a factor that is affecting the distribution of the genome in the nucleus. And I don't know, I don't have an answer for that, but it will definitely be interesting to look into that. Yeah.
So how much strain variability have you found in your experiments? My impression is that you would use what I'll call inbred lines that are all, they have a homogeneous background. Have you looked at different strains, the response of different strains to TBT and arsenic?
So I haven't done that experiment yet, but it is part of a proposal that is pending. So hopefully it gets funded and we can do it. Yes, basically we have been working with a mouse strain that is called C57BL6 mice. And it is an inbred line. So as you were saying, they are genetically the same, all the animals. There is a lot of discussion about whether we should use an inbred line versus an outbred line. That the outbred line indicates that there is a variation among individuals. And I am definitely interested in that. And as I said, we are waiting to hear about funding to that experiment. And this is important also if we want to answer questions related to human populations, because we are not genetically identical. Right. So the work that we do in mice has many limitations. One of them is the fact that they are mice and not humans. But another factor is that we are not accounting for the genetic diversity that we are finding in human populations. So we are actually hoping that we can get that done. Oh, sorry. Okay. Well.
Did you get to see Carlos Jr.?
No, Carlos Jr. is at home.
Yeah.
That's not a problem. Right. Yeah.
I was at a workshop with Marcus Pimbry, who was a real leader in some of the analysis of the Swedish populations, where they knew that there was low food availability back in the early 1900s, and then they tracked health effects on the grandchildren of sort of like your Dutch hunger example. And he was really careful to point out that it is so hard to do controlled experiments. You're trying to introduce a variable at a time, but there are so many confounding variables that we have no control of. The mouse colony conditions, the temperature. It's really challenging to think about how to keep all of those variables to a minimum so that you can just do TBT exposure or arsenic exposure or exposure analysis.
I completely agree with you. And actually, I'm going to link back to the microbiome question. There are actually changes in microbiome found in the same mouse strain that you can use, depending on whether they are located in one room versus a different room in the company where we buy the animals from. So just that variability is introducing confounding factors. Not to mention when we are talking about different facilities that are located in different cities. And so that is definitely important. However, I have a comment about that, and I know that it might be controversial, but if the effect that we are seeing really depends on controlling every single factor that can be playing a role, that means that the effect is not that strong. It's not that so I think I tend to have like a relaxed attitude when it comes to confounding factors. I think it is important to know they exist. I think it is important that we identify them, the potential confounding factors, to make sure that we make the right conclusions out of our studies. But I try to relax a little bit because it is impossible otherwise. It's just,
Yes, I agree. I'm whizzing through the Q&A box, which has gotten quite long. I didn't understand the link to obesity. Can you please elaborate a bit on why there is a trend in global obesity?
The link to obesity, like my study, like the sorry, I know, Susan, that you are not the, the
I'm not the
that is
question asker.
so it will be like a link with obesity. So exposure to environmental factors and how that can be affecting obesity. I mean, if that is the question, which I'm not sure, but if so, what we are actually trying to figure out is that connection between the exposure to environmental factors and obesity. And going back to the confounding factors, it is very difficult to do in human populations when populations are exposed to many different things, and depending on location, depending on the moment in life we are exposed to those factors. So there is still a lot of work that needs to be done, and we are working on that. And actually there is a lot of communication between epidemiologists and basic researchers to actually narrow down the mechanisms of action of the effects that we are seeing in human populations to try to explain certain diseases. So I think, we need to keep working.
Well, on that note, I agree with that. And we're approaching the hour, at which time I'm going to turn this back over to George. And I just want to first give you my heartfelt thanks for this great seminar, really thought-provoking, fascinating, important, lots of great questions. I thank all the participants for asking them. And thank you, Raquel. And I'll turn it back over to George to wrap this up.
All right. Well, thank you, Susan, for moderating this and to keep everything on track with time and everything, and everybody for attending and for making these great questions.
Well, thank you, Raquel and Suzanne, for sharing this important research with us tonight. Please join us for our next Kraw Lecture, May 18th, with astrophysicist Professor Neil Imara, telling us how to build a star. Good night, everyone.
Good night.